It's my great pleasure to welcome you at the 62nd annual meeting of the Society of Psychophysiological Research. And I'm very happy that we have a face-to-face meeting after 2 years of virtual meetings again. And I'm also happy to see, or have seen, I can't see anything because of the lights actually, to see familiar faces and many attendees being here. At the same time, this is a fully hybrid meeting. So also a warm welcome to all remote attendees. And we have invested quite a lot of time and effort and also budget to make this hybrid experience possible. So it's very important to us that you give us feedback in the end. What worked out, what didn't work out, what you would want to keep in future meetings, what we could drop, and so on. So we want to learn from this experience because it could be a way of also making SPR meetings more accessible to people in general if we have more online content and streaming. So I would like to thank many people who have made this conference possible. First of all, the program committee and its chair, Jan Wesse. So he has put up, or they have put up a great program. Also, I would like to thank our secretary, Becky Silton, the officers, the board of directors, and of course, a big thanks to the Rees Group, and particularly Jane Shepard, without whom this wouldn't have been possible at all here. And thanks to you that you're here to present your work, to engage in discussions. So let me welcome you again, and I declare this meeting open. And before we start with the first keynote lecture, I have to make some announcements. So first of all, I would like to remind you that there is a poster session. The second poster session tonight is just next door in Regency Ballroom C at 6:10 p.m. And then one thing that everybody should know who is speaking at a symposium or the big idea session, so please make sure to visit the speaker ready room at least 2 hours before your session, such that you can set up your slides and everything. Then I would like to remind you that the virtual poster session is scheduled tomorrow at 11:30 a.m. So please log into the virtual platform and then click on the abstracts page and visit the virtual posters and authors. And all students are invited to the Student Social on Friday evening at 10 p.m. at Fable Diner and Bar. And beforehand, please stop at the Committee to Promote Student Interest meeting at 8 p.m. That's in Tennyson on the 4th floor. So without any further ado, I will hand the word over to Jan, who will present our first keynote speaker.
Hello, everyone. I'm the aforementioned Jan. Thanks for being here. I'm here on behalf of the program committee, and I am extremely honored to introduce our first keynote speaker of the 2022 meeting. It's Dr. Stephanie Jones from Brown University. Stephanie Jones received her, she's a lifelong New Englander, actually, as we recently noticed, or I recently noticed. She received a BA and an MA in mathematics from Boston College, and then went on to earn a PhD in mathematics from Boston University. And then she spent about 10 years at Harvard Medical School in different positions, ranging from postdoc to faculty. And since 2018, she's been an associate professor of neuroscience at Brown. Stephanie has received many accolades and awards, including the NIH Brain Initiative Award and the Biomag Mid-Career Award. But since I imagine that there's at least some of you in here that are going to be exposed to Dr. Jones's work for the first time, I actually wanted to briefly share an anecdote of how my lab and I learned about her work for the first time and what happened. This was in 2016. Steph's lab had just put out a phenomenal PNAS paper that I would recommend everybody read, and I was asked to comment on it for a media publication, so I read it. And now I don't have Stephanie Jones's brain, so it took me, a more limited capacity, so it took a second. But once it clicked for me and I really understood what I was reading, I think, it really led to a fundamental reappraisal of the way that I thought about the EEG and LFP and intracranial data that we often record. So much so that I took it back to the lab, I made everybody read it, I was gung-ho and super excited about it. And really what we've done is we've taken what we've learned from Dr. Jones and we have applied it without collecting any new data to all of the data that we've had lying around in the lab. And so far that's produced, several JNeuro eLife papers, $2 million grant, all because of what I learned the first time I listened to, I learned of Dr. Jones's work. So while I'm not guaranteeing that will be happening for everybody when that happens to you, if that's what happens to me, then just imagine what could happen to all of you when you learn about Stephanie Jones's work. So without further ado, I would love to hand it over on behalf of the program committee to Dr. Jones from Brown University.
Moved it. Hello, everyone. Well, wow, thank you, Jan. That was, I think, the nicest introduction I've ever had, and I'm thrilled to hear that we are influencing your work as you are influencing ours, and I'll talk a little bit about a collaboration that we have ongoing now. And thank you all for being here. I'm really thrilled to have the opportunity to describe our research program to this society, and to get the feedback from you all on some of the things that we're doing in the lab. And I'm going to specifically be talking about a software that we've been developing called Human Neocortical Neurosolver, and it's for cellular and circuit level interpretation of human EEG and MEG signals. So I'd like to begin by describing the challenge in human neuroscience that we're trying to address with the development of this new Neocortical Neurosolver software tool. We all know that human EEG and MEG are really powerful technologies. They give us direct correlates of human information processing with millisecond resolution. But there's a big downside in that what we're recording is considered macro scale activity. It's generated by large ensembles of neurons, and it's still really difficult to infer what's going on at the underlying cellular and circuit level to create these signals. And this circuit level understanding is really critical if we want to use EEG to develop new theories about information processing, or if we want to use it to develop new therapies for neuropathology when EEG signals are disrupted in pathology. Now, recording with invasive recording techniques and all the cool neuromodulation with optogenetics and everything we can do in mice is obviously ideal for dissecting this circuit level activity, but there's obvious challenges in addressing that and applying these techniques in the human. And so to bridge this gap, we've been applying computational modeling methods. And there's two types of modeling. The first type of modeling links the extracranially measured signals to the underlying electrical currents in the brain that create these sensor signals. And so we're estimating the location and the time course of these big electrical currents in the brain that create the EEG or MEG. From there, we need to connect these electrical currents to the cell and circuit level origin. And for that, we use computational neuromodeling, where we simulate the electrical activity of the individual neurons in the circuit. And with this type of modeling, we can have specificity both at this circuit level, this micro scale level, and at the macro scale recording level. I think I just lost my pointer. Oh, it's there, but I'm not seeing it. Do you have another pointer?
You guys have another pointer back there somewhere?
In any case, I'm just going to keep going on. So this type of modeling has been the primary focus of my research program. And we've recently turned this model into a software called Human Neocortical Neurosolver, or HNN, that we're developing for the community to start to be able to develop and test predictions on the cellular and circuit level origin of their signal. And so in my talk, I'm going to start by just giving you some background on the development of this tool. And then I'm going to show you how we've applied it to study one of the most commonly measured macro scale signals, 15 to 29 hertz beta rhythms. And I'm going to focus on some studies in my lab where we've been studying the role of beta in sensory inhibition. And in the end, I'll briefly describe some other applications to study healthy and pathological cognitive processes. So to begin, how do we begin to develop a model that could connect these extracranially measured signals to what's going on at the cell and circuit level activity? And so as I've mentioned, the first thing that we do is we apply these inverse solution methods. And what these do is they take the sensor signal and they estimate, now it's working, so here we go, the location and the direction of these big underlying currents. We call these primary currents. Now we know that these primary currents, these big electrical signals, are generated by the intracellular current flow in long and spatially aligned cortical pyramidal neuron dendrites. Essentially, the length and aligned orientation of these dendrites creates this whomping electrical signal that you can record outside of the head with EEG and MEG. And so knowing that this is where the signal comes from, there are some key canonical features of cortical circuitry that we have to include in our model to get at the cell and circuit level generation. The first thing we have to consider is that the cortex is a layered structure. We have pyramidal neurons in the supragranular and infragranular layers that have these long apical dendrites that extend through the layers. These are synaptically coupled to other inhibitory neurons and excitatory neurons. This cortical circuit doesn't sit in isolation, but it's constantly receiving input from other parts of the brain. And there are two primary pathways of information flow into the cortex. One pathway comes from the lemniscal thalamus. This relays sensory information through the body up to the thalamus and into the granular layers of the cortex, where it then propagates to these pyramidal neuron dendrites. The other type of input comes right into the supragranular layers. This is the input that can come from higher order cortical areas, or there are these thalamic nuclei considered non-specific thalamic nuclei that project right up to the supragranular layers. And then this primary current dipole can be calculated from the net intracellular current flow in these pyramidal neuron dendrites. So we just sun up these electrical currents. And with this construction, the units of measure that come out of the model are current times distance, nanoampere meter. That's the same exact unit that you get when you source localize your data. And so you have one-to-one comparison between the output of the model and your source localized signal, which can be really powerful in generating predictions about where these signals come from. And we know something about the size of the network that contribute to some of the most commonly measured EEG signals, including event-related potentials and low-frequency brain rhythms. ERPs tend to be on the order of 10 to 100 nanoampere meters. They're thought to be generated by on the order of tens of thousands of synchronously firing pyramidal neurons. Low frequency oscillations are even higher in amplitude. They tend to be on the order of 100 to 1,000 nanoampere meters, and they're believed to be generated by on the order of 1 million pyramidal neurons in the subthreshold regime. So this is obviously a reduced representation of all the fine details of the cortical circuit, because when we record at the macro scale outside of the head, we don't have access to those finer details, but we can still say something meaningful about the origin of our signal from this canonical structure. And so this is the model that's included in our software. And just to give you a little bit more detail, we simulate the electrical activity of the neurons using what we call multi-compartment pyramidal neurons. We have compartments for the soma and the dendrite. The inhibitory neurons, we model with what we call single compartments. They're just point neurons. They don't contribute to the signal that we're recording, but they're very important for the dynamics that this network creates, so they're included in our model. We have GABAergic and glutamatergic synapses between these neurons. We simulate the voltage in each compartment with what's called a Hodgkin-Huxley Hodgkin-Huxley dynamics, and then we get the intracellular current flow just from regular old cable theory. And then again, the primary current dipole that we're going to match to our data is just summing up the electrical currents in these pyramidal neuron dendrites. And I've mentioned that it's important to consider that this cortical unit is receiving input from other parts of the brain. In our model, we're not simulating the whole brain. what we do is we generate trains of action potentials and predefined patterns that activate this cortical column through these projection patterns. And so, for example, I can create a pattern of spikes that represents feed-forward drive that's going to activate this local cortical column through this feed-forward projection pathway. We also call it proximal drive because it effectively hits the proximal dendrites of our pyramidal neurons. This input will come up, it'll activate synapses and can drive current flow up the dendrites. That would create an upward deflecting signal. I can also create a train of action potentials that represents feedback input. And this is going to activate synapses on the distal dendrites and can push current flow down the dendrites to create a negative deflecting signal. And it's a combination of these inputs with the local circuit dynamics that create this dipole signal that I'm going to compare to my data. Now this is a reduction of the full model. The full model represents a patch of cortex. It has a scalable number of pyramidal neurons and interneurons, and it's representing an area of cortex in the brain. And we're specifically designing this software to simulate some of the most commonly measured signals from a single brain area, including event-related potentials and low-frequency oscillations in the alpha, beta, and gamma bands. Now, this is a really complex large-scale model. There's thousands of differential equations. We are designing the software with best practices in open source software design so that if you know how to build and work with neural models, you can help us develop it and expand it. But many in the community don't know how to begin to work with these models. And so what we've done is we've embedded this model in a graphical user interface. And we created a website where we distribute information about the model. And importantly, we have tutorials to tell you how to begin to study event-related potentials and low-frequency rhythms in the alpha, beta, and gamma bands. These tutorials build from our own prior published work. So we give you the data, we give you the model parameters, we teach you what we did, and then the idea is once you know how we did it, you can then apply it to your own data. And again, we are trying to do this with best practices in open source so that the community can help us expand it and develop it, and we already have some community development. And so this is a figure of the graphical user interface, and you can see at the top, we have a representation of the underlying model. We show the local network connectivity and the input that you can put in through these proximal and distal pathways. And when you are using the software, the name of the game is defining the input that represents your simulation experiment. And I'm going to show you an example of that now from work in my lab. So I'm going to show you how we've used it to study the mechanisms and meaning of these 15 to 29 hertz beta rhythms. So in my lab, we study somatosensory perception. And we do these very boring studies where we give subjects a brief tap to the finger and we ask them, did you feel it or didn't you feel it? And we titrate it so they feel it half of the time. And then we look at the brain response and correlates of perception. And what I'm showing you here is we've applied our inverse solution technique to isolate the contribution of the signal to from the hand area of S1, contribution to the signal from the hand area of S1. And I'm showing you here just the spontaneous signal from S1 before we tap the finger. And you can see there's this large amplitude, low frequency oscillation that when we put a frequency filter on it has this very strong component in the 15 to 29 hertz beta band. But at any moment in time, beta power is changing. And so the question we wanted to know was, does pre-stimulus beta power impact the ability for the subject to feel that tap to the finger? And the answer to that question is yes. The more beta power you have in the pre-stimulus period, the less likely you are to feel that tap. And so what I'm showing you here is we've taken this 1 second pre-stimulus period, we've averaged across time, we've averaged across the band, and we've sorted from low to high power. And then we're plotting the detection rate as a percent change in hit rate from the mean. So if it's lower, you're more likely to detect, you're less likely to detect, higher, you're more likely to detect. And so the more beta you have, the more likely you're going to say, I didn't feel the tap to the finger. And so in this way, beta seems to be a signature of inhibited perception. And there's several other examples in the literature that suggest that in general, beta may be playing an inhibitory role. We've done some studies where we've looked at shifts in beta rhythms with attention, and we find that beta rhythms go down, and they go down in coordination with frontal cortex when you're paying attention, suggesting that they're getting out of the way for optimal information processing. We and others have observed that beta rhythms increase in sensory motor cortex with aging, and that this maps onto a decrease in tactile and motor acuity. Jan and others have shown that beta rhythms in prefrontal cortex are an index of motor cancellation in stop signal tasks. They're also overexpressed in motor cortex in Parkinson's disease. There's lots of evidence that beta is for some reason inhibitory to processing. And so the question we wanted to know was why? What is it about the mechanisms that create beta that cause it to be inhibitory to function? And so for this, we turn to our computational neural model, HNN. But good representation of this data. And so across time, we average across subjects, but the brain isn't averaging. Anytime I tap the finger, we're not averaging. And so we want to understand, well, what does it look like in unaveraged data? Once we know the answer to that question, we can ask, well, what features of this unaveraged data are contributing to these functionally relevant difference in power that we see? The more power you have, the less likely you are to feel the tap to the finger. Where is that coming from in the signal? Then we can turn to our model and ask, well, what are the circuit mechanisms that create these differences that we found? And finally, how do these mechanisms translate to inhibited function? To, I didn't feel the tap to the finger. And I'm going to give you the punchline of what we've discovered by answering these questions. And what we've come up with is this new theory on the circuit level dynamics that are creating beta. And this theory predicts that beta is decreasing the saliences of sensory information by recruiting layer-specific inhibition. And this inhibition decreases the relay of sensory information out of the cortical area, and the subject says, I didn't feel the tap to the finger. And so I'm going to unpack how we've come to that theory. And I think this is the theory paper that inspired Jan. Okay. So the first question, what does beta look like in unaveraged data? So what I'm showing you here is we take this pre-stimulus data before we tap the finger, and I'm just showing you 10 example epochs of 1 second of pre-stimulus data. You see the waveform and then the time frequency representation. And what you see very clearly is that in unaveraged data, these beta oscillations are very transient. They tend to last less than 150 milliseconds. We call them beta events. Now, because in the frequency domain, the signal is always positive. When you average this, these beta events accumulate without cancellation, and you get this perception that there's this continuous ongoing oscillation. But that's just an artifact of averaging these transient events that we see in our non-average data. We've seen that this is a very reliable phenomena. What I'm showing you here is data from our human tactile detection experiments, and this is MEG data, but it looks exactly the same if we do it with EEG data as well. And on the right, I'm showing you local field potential data from the barrel cortex, the vibrissa somatosensory cortex of mice from my colleague, Christopher Moore at Brown, where they're doing tactile detection of vibrissa deflections. And we've quantified things about these beta events. We've looked at, well, how many of them are there in a 1 second period? We've looked at their power, we've looked at their duration, their frequency span, and we find remarkable homology across tasks and species and recording modalities that suggest that there's some very robust underlying phenomena. And I just want to point out here that we're now sharing some of our tools that we're using to quantify these spectral events in different frequency bands. And so that's an open source tool that you're all welcome to use and apply to your research. And this transient beta event phenomena has now been shown in lots of studies, including studies that I mentioned by Jan, where they're looking at the frontal cortex signal in these stopping action paradigms. And these beta oscillations are coming out as these brief transient events. It's been shown in motor cortex, it's been shown in subcortical structures, and so again, this seems to be a very robust, reliable phenomenon. The other thing we've done is we've looked closely at the shape of the signal that's underlying these high power events. And so what I'm showing you here is here's the raw time trace from the 1 second period before we tap the finger. This is the corresponding frequency domain. We find a high power beta event, and then we go back to the raw signal and we align them in time. And so what I'm showing you here is 50 high power beta events overlaid, and on the bottom I'm showing you that from 10 different subjects. And we quantified some things, but you can see very clearly that this is defined by this prominent negative deflecting peak that lasts 50 milliseconds. Now, when you put a frequency filter on that, it creates a high power beta event. But the reason that it's beta is because you had an event that lasts 50 milliseconds. This is not a sinusoidal oscillation that has a 50 millisecond period. And understanding this shape is really important when we want to understand what are the circuit mechanisms that create it. We've also seen this shape now in frontal cortex and motor cortex. It also perhaps is generalizable. Okay, so we've answered the first question. What does beta look like? Beta events are transient, and they typically last less than 150 milliseconds, and they have this stereotypical waveform shape. So now, what features underlie functionally relevant differences in average power? Remember, the more power you have, the less likely you'll feel the tap to the finger. Now, when we first started this study, we automatically thought, oh, if I have more power, I have a higher amplitude oscillation. It must be that there's a bigger portion of the network that's contributing to this phenomena. But now that we know that these oscillations are transient, there are several things that can contribute to high power. It doesn't have to be a higher amplitude oscillation. It could be that on trials where we have low power, we have one event, where trials where we have high power, we have two events of the same amplitude. But you average this, you get high power, you average that, you get low power. It could be that the amplitude of the events is indeed higher, right? And so maybe we have one event in each epoch, but this one has more amplitude, and when I average, I get higher power. It could be that the duration of the events is changing, such that when you have longer events, you get higher average power, or it could be that the frequency span is changing. Any one of these could underlie the difference in average power that we're seeing. And it turns out, when we correlate the number of events, their amplitude, their duration, or their frequency span with the average power in the pre-stimulus window, and this is just a Pearson's correlation plot, box and whisker plot over 10 different participants, you can see very clearly that it's the number of events that's dictating the power of that signal. And here's just the raw data. And again, you see it clearly. Here I'm sorting the power and then looking at the number of events or their amplitude, their length, or their duration. And so when I have high power, it's because whatever this beta event process is, it's happening more often. Now, does that translate to what's happening with that's impacting function, right? Remember, when we have higher power, we are less likely to feel that tap to the finger. Is that also because I have more events? Or maybe what matters for perception is that the events have a different amplitude or a different duration. And so we asked that question. Oh, and I just want to show you again, there's consistency across different experimental paradigms, different recording modalities, a very robust phenomenon. So next we looked at how do the number of events, their amplitude, length, or duration correlate with this hit rate, which is again the percent change in detection from the mean. And so I'm looking at sorting either the number of events from low to high, their amplitude, their duration, or their frequency span. And this is higher means you're more likely to detect, and lower less likely to detect. And you can see very clearly that the only relationship is with the number of events. We also pooled over hit and miss trials and found that there are more events on trials where the subject said, I didn't feel the tap to the finger. We saw a similar phenomena in our attention tasks, such that when you're saying pay attention, you're decreasing the number of events. And we saw something very similar in this mouse vibrissa detection task. So again, very reliable. The other thing we looked at that I'm not showing a figure of is the timing of the events relative to that tap to the finger. The closer an event occurs to the tap to the finger, the more likely it is for you to say, I'm not going to feel that tap. And so what features underlie these functionally relevant differences in average power? It's the number of events is greater during non-detected, non-attended trials, and non-detected trials have more recent beta events to the tap to the finger. So there's some process in the brain that when I say pay attention, you decrease the probability of this process happening, and that leads to you having improved perception. Okay, So now we can turn to our model. Now we know what we're modeling, right? We're modeling a signal that looks like this. And we're going to use this patch of cortex with these input patterns that I can create to study where does the generation of this come from. Now this is a really large scale model. There's thousands of parameters, and I often get the question, well, how do you constrain the madness, right? Is there a constraint that you can put in this without varying all the parameters? And in fact, there are several constraints that you can put in. There is structure here, there are biophysics here that dictate what will come out of this model. And so the first thing that we did was we built the individual neurons. We fixed their morphology, we fixed their physiology, and we tuned it so that they reproduced realistic spiking patterns to injected current. Then we fixed all of those parameters. Then we connected the local network together. There's a lot of literature out there about how the laminar structure of the cortex is wired together, particularly in the somatosensory cortex, which is where we started. And so we built that in and we fixed those connections. The only parameters that we started to tune when studying the origin of these beta events were the timing and the strength of these exogenous inputs that are going to activate the synapses in the local network. And through a long history and many years of trial and error, we came up with the following prediction: that beta events, and I should say I had a bias as to how beta events were going to be generated in the network, because I had, for my PhD, studied some oscillations and had preconceived notions about how beta oscillations should come about in neural circuits. And then we put in this structure, and I looked at my data, and I said, that mechanism's just not going to work. And so what might work? And what we came up with was this. Beta events are emerging from the integration of two bursts of input that come into the network in these proximal and distal projection patterns, such that this distal input is stronger and it lasts 50 milliseconds. And so what happens in the model is this is a histogram of the spikes that are going to drive the network through these projection patterns. And so this is the proximal spike histogram and the distal. And what happens with these proximal spikes is they come in and they push current flow up the pyramidal neuron dendrites. And if I didn't have the distal drive, I would just get a positive deflecting signal. But at the same time, I have this strong distal drive that's coming in and it's pushing current flow down the dendrites, and it lasts 50 milliseconds. And now I get this waveform that looks just like my beta events. This very nicely reproduced all these features that we quantified in our MEG data. But because this is a model and we have all this microcircuit level prediction, we could go in and we could test this model with invasive recordings in mice and monkeys. And fortunately, through collaboration with Chris Moore and Saskia Hagens and Charlie Schroeder, we were able to test some of these predictions. And we have some initial validation that this prediction might be true, both from laminar recordings and current source density in mice and monkeys. And more recently, there's a technique to do laminar resolution MEG in humans. In a recent study, has looked for evidence that this mechanism might be occurring during beta events and found that it could be true. So this is really exciting predictions coming out of the model. Okay, so what are the circuit mechanisms underlying these differences? Beta events emerge from a broad proximal excitatory drive that's simultaneous with a strong distal drive that lasts 50 milliseconds. And that generates your beta event. So how does this mechanism translate to inhibited perception? If I have one of these events, I'm not going to feel the tap to the finger. If I have it before I tap the finger. And so what we did was we took our model and we said, well, let's simulate an event and let's simulate a tap to the finger. And so first we looked at our data, right? What happens when we look at the evoked response on detected and non-detected trials? Trials where the subject said, I felt the tap and I didn't feel the tap. And what we found were there were some quantifiable differences, and that on detected trials, which is the dark blue here, we had this longer duration negative deflection, and the amplitude at the end was higher compared to non-detected trials. We also sorted over, did you have a beta event in the pre-stimulus period, or didn't you have a beta event in the pre-stimulus period? And that's what I'm showing in the light blue and the light red traces. And you can see there's markable similarity whether I sort over beta, having a beta event, or whether I sort over perception. And it suggests there's a causal relationship between having a beta event, having an impact on the tactile evoked response, and that having some kind of influence on your perception of this tap to the finger. And so we're going to turn to HNN and ask, how did these beta influences influence the evoked response? And why does this correspond to I didn't feel the tap to the finger? And so we're going to simulate the S1 evoked response, which I'm going to show you in a minute. And we're going to do that with and without a simulation of this beta event mechanism and see if we can reproduce this effect and if it can tell us anything about the underlying neural dynamics of what's happening. And so I'm going to step back here and show you how we model an evoked response. And this was actually the first study that we did with the HNN model. We were looking at tactile evoked responses. We saw this difference with perception. And so we went to our model to simulate the evoked response. So where do we begin? Well, there's a large theory out there that this evoked response has been studied with invasive recordings for decades. And so we went to the literature and said, what do we know about where these ERPs come from? And so the first thing I want to show you, this is our software. We load in our evoked response data. This is the data source localized to the hand area of S1 that you've seen. An important thing to point out is that we were also able to confirm that this prominent peak right here happening at 70 milliseconds corresponds to current flow down the dendrites into the cortex. And we get that information from our source localization. And so we went to the theories on where do ERPs come from, and the literature suggests the following. And I referenced just a few of the papers here, but there's a long literature of this. The S1 evoked response is believed to be generated by a sequence of activation of that local network. After you tap the finger, input comes in, and approximately 25 milliseconds through this feed-forward pathway, it activates the cortex. Approximately 70 milliseconds after you tap the finger, it goes somewhere else and comes back in a feedback projection pattern. And then you have a re-emergent thalamocortical loop of activity about 100 milliseconds after, and you get this re-emergent thalamic information. And so we can build this sequence of input into HNN and test, does this hypothesis that the sequence of this S1 activation, can it generate this S1 ERP? And so what did we do? We simulated feed-forward input at 25 milliseconds, feedback input at 70 milliseconds, and another feed-forward re-emergent thalamic input at about 125 milliseconds. This input is going to come in, it's going to activate the local network, and it's going to produce some kind of response in my pyramidal neuron dendrites. And so when we run this simulation, you can see that it very nicely reproduces the S1 ERP. Now, the literature guided us to where to begin, right? What is going on? And so we knew, well, let's start to drive it with this sequence of input. But we spent a lot of time tuning the timing and the strength of these inputs in order to get an accurate representation of this signal. And that led to all these microcircuit predictions that we could go in and verify in the literature. And so that now that we have a nice representation of the ERP, we can start to develop and test hypotheses about where differences for different ERPs come from. And for us, we're going to be looking at the ERP difference with and without a beta event in the pre-stimulus period. But before I do that, I just want to show you briefly that HNN has shown that this sequence of input can reproduce ERPs in other sensory areas. And so what I'm showing you here is our S1 evoked, I'm looking down there. The S1 evoked response from a brief tap to a finger. This is the A1 evoked response from a brief presentation of a tone. And this is the V1 evoked response from a brief flash of a checkerboard. And you can see there's differences in these ERPs, but they have some common structure. They all start by going up, and then they have this large deflection going down, and then they have this re-emergent upward deflecting peak. And so in H&M, what we showed is that the same sequence of input, proximal, diximal, proximal, can create the ERP. And again, we're tuning the timing and the strength of these drives to get a representation of the signal. But what this suggests is that the macro scale, what we're recording, is dictated by commonalities in cortical structure and in these input patterns into the cortex when we give a sensory stimulus. Okay, so now back to the study at hand, which is we have our ERP. We want to understand how do beta events in the pre-stimulus period impact the filtering of this incoming information and impact the ERP. So here again is our MEG data. I've just now overlaid everything rather than showing it on 2 plots. And we ask, how do beta events influence the evoked response? And why does this correspond to non-detected trials? And so again, what we're going to do in our model is we're going to simulate our beta event mechanism, and then we're going to simulate the tap to the finger, and I've now showed you how we do both of those things. And remarkably, when we do this in the model, it reproduces these signature differences that we see in our data. And I always like to stop here in the talk and say we actually did this in the model first. We had a mechanism for the ERP. We had a mechanism for a beta event. We said, what's going to happen if we put these two things together? It looked like the figure on the bottom, and then we went and looked at it for our data, and it was there. And this was one of the moments as a modeler where you just get the chills, and you're like, there's something generative. You know, models are always wrong, but there's something right in what we're doing in our H&N model. Okay, so just to give you a little insight, why does this happen, right? Why is it that when I have a beta event, I get the blue curve, and when I don't, I'm sorry, I said that backwards. When I don't have a beta event, I get the blue curve, and when I do, I get the red curve. What is happening is the following. Remember, beta events are created by this strong distal drive. This distal drive is recruiting a lot of inhibition in the supragranular layers. Remember, the model does have inhibitory neurons. That inhibition shuts off the effect of this feedback drive that creates this downward deflecting signal, right? I have a lot of inhibition, I tap the finger, the feedback comes in, and it gets shunted. When that happens, you get less firing in the pyramidal neuron populations. And so you get a decreased amplitude of the red curve in the later time points. If you have decreased firing in these pyramidal neuron populations, these are the ones that talk to the rest of the brain, then you get a decrease in relay of information out of the primary somatosensory cortex, and the subject says, no, I didn't feel the tap to the finger. And so again, beta events are recruiting inhibition in the supragranular layers. That shunts the impact of this tactile evoked response from getting in, and if it can't get into S1, it doesn't get out of S1 to tell the rest of the brain that you felt it. And just to show you that in a little bit more detail, remember this is a model. I can look at anything in the model. And so what I'm showing you here is the spiking activity in the different neural populations when I simulate the ERP with a beta event and without a beta event. And so with a beta event, you can see before the tap to the finger starts, which is right here at 25 milliseconds, this is when it hits the cortex, that beta events recruit a lot of layer 2-3 inhibition. And so then, when this sequence of feed-forward, feedback, feed-forward comes in, it gets shunted. And we don't get any pyramidal neuron firing. And so again, if you don't have pyramidal neuron firing, they don't get out, and the subject says, I didn't feel the tap to the finger. So, how does these beta mechanisms translate to inhibited perception? Beta is decreasing the salient sensory information via recruitment of this supragranular inhibition that's decreasing the relay of sensory information. This right now is a new prediction from the model. It hasn't been validated. We're fortunate to have some collaborators that are starting to test some of these predictions. But it's just an example of the power of how we can use the model to study why do these signals matter for perception and cognition. Okay, so in the last few minutes, how am I doing? Yeah, am I pretty good? All right, I'm doing great. I just want to briefly describe some other applications of H&N to study healthy and pathological cognitive processes. And the first study that I want to highlight is one that's a new collaboration with Jan Wessel and his prior graduate student Darcy Deisberg, who I am thrilled to say is now a postdoctoral researcher in my lab, starting just this month. It's wonderful to have her. Darcy is giving a virtual poster on this work, and so I encourage you to check out that poster to learn more details about what I'm going to tell you. And so what we were trying to do with H&N in this study was use it to see if we can gain some mechanistic insight into signatures of motor inhibition. And we specifically wanted to test a prediction that comes out of the cognitive behavioral theory of the horse race model of motor inhibition that suggests when you have to stop an action, there's a race between go and no-go processes, such that successful stops result from a faster stop process. And so we wanted to see, can we use HNN to gain insight into whether this faster stop process may be happening? And so in this stop signal task, subjects are seated looking at a screen, and every 3 seconds an arrow appears that cues the participant to press a button with the finger from the hand that the arrow is pointing to. On a third of the trials, the arrow turns red, and that cues the participant to stop the action. And the timing of the arrow is titrated so that they successfully stop half of the time. And what we can do is we can look at the ERP from the frontal cortex, and here we're looking at the FCZ electrode, and we're aligning it to this stop signal. And what I want to point out is that this very prominent peak that's called a P3 is earlier on successful stop trials. The amplitude is the same, but it's occurring earlier. And so we wanted to see, can we apply HNN to test if we can link this P3 observation to the neural mechanisms that are reflective of a faster stop process on successful stop trials. And so the first thing we did is we went into HNN and we used knowledge about the sequence of input that can generate an ERP from our prior studies. And we built that in and simulated the successful stop trials. And so here what I'm showing you, again, is this histogram of input. Now it's all on one axis, but we have proximal, distal, late proximal drive. We tune the timing and the strength of these inputs to get an accurate representation of our stop data. So here the data is shown in blue, and the model output is shown in light blue. And so you can see with this sequence of input, we can reproduce this stop process. And importantly, this P3 peak is coming from this late proximal drive, which represents a thalamicortical input that's occurring at approximately 300 milliseconds. We pulled these spike times from a Gaussian with a mean and a standard deviation, and the mean here is at 305 milliseconds. That creates this positive deflecting P3 peak, which peaks at the same time of our data is 312 milliseconds. So assuming that this is regulating the stopping process, this thalamicortical input, we used HNN to test the predictions that the later P3 in failed stop trials can be accounted for by delaying the onset of this thalamicortical drive. And indeed, when we delay the onset of that input, here we're delaying it by 20 milliseconds. And so I'm taking this last proximal input, shifting it over by 20 milliseconds. That was able to reproduce the shift in the P3 timing while maintaining the same amplitude. I'm not changing the strength of the drives at all, I'm just shifting the timing. And so this lends support for the idea that there is a faster stop process in successful stop trials. Now, we tested several alternative mechanisms. Well, could we change of the synaptic strength? Could we change other properties in the model and reproduce this phenomena? And several other alternative mechanisms failed to reproduce this. Again, lending support for this successful stop process. So lastly, I just want to give you some examples, other examples of where HNN has been applied. We've used HNN to study auditory processing. We've looked at the circuit origin of hemispheric differences in auditory evoked responses. We've used HNN to study the impact of non-invasive brain stimulation on circuit dynamics and how they're reflected in a change in the EEG signal. Other groups have used the model to study changes in low-frequency beta and gamma rhythms in children with autism spectrum disorder. We have an ongoing study with my collaborator, Dr. Sharif at Rhode Island Hospital, to look at the circuit effects of ketamine for depression based on EEG correlates of symptom improvement. And I have a new study looking at biomarkers of Alzheimer's disease and some things that are predicting the transition from mild cognitive impairment to Alzheimer's. And here we are focusing on these beta events and resting state oscillations. And so that's just a handful of the studies that we're doing. I encourage you to check out the publications and talk more to me if you're interested and learning more. And so that I'm going to conclude. And so HNN's foundation is a neocortical column model on the thalamocortical and cortical drive that simulates EEG and MEG primary current dipoles based on their biophysical origin. We've embedded this model in a user-friendly graphical user interface, and we are trying to teach and train the community how to use it to study the origin of their data. HNN combined with MEG and invasive recordings in animals revealed this novel prediction on the mechanism and meaning of these beta events and suggests that beta is decreasing the salience of sensory information via layer-specific recruitment of inhibition in the supragranular layers. And hopefully I've convinced you that HNN can be applied to uncover circuit mechanisms underlying EEG and MEG correlates of healthy and neuropathological processes. Ultimately, we hope that it will be used to develop treatments when these signals are disrupted in disease states. And so with that, I want to thank you all and thank my collaborators and funding sources. I have an amazing lab at Brown, and everybody's contributed to this research in a meaningful way. The HNN software is being developed by a multi-institutional team at Brown, the Martino Center at Mass General Hospital, and Yale University. I've been really fortunate to develop a network of collaborations that allow us to do this interdisciplinary research. And so I'm grateful for all of our collaborators. And so that, I'm just going to leave you with our conclusions and take any questions that you have. Thank you. There are a couple microphones if there are any questions.
Hello, that's beautiful work.
Thank you.
Bridging from cells to ERPs is a wonderful thing. So it's a wonderful advance. One thing I'm wondering about is how the model handles or why it can ignore really the geometric aspects of the cortex and the orientation of dipoles. You see it conventionally, there's been a lot of debate about which direction, how is the dipole oriented, how does that translate to scalp recordings of ERPs and the polarity of the ERPs. So how does the model get past that?
And we don't ignore it, actually. It's a very important piece of the puzzle in determining what are the circuit mechanisms. The first thing that we do with our source localization is we maintain the orientation. And so we know at certain periods of time whether the signal is going in or out of the cortex. I don't know if you remember when I showed that S1 ERP, there was that little picture and I said at 70 milliseconds, current flow is going into the cortex. And so that information is important when we want to understand it with our model. Should I be simulating something that goes up or down the dendrites? And so the first step in using HNN is to understand your signal. And that means understand your signal at the source level and whether current at certain points in time is in or out of the cortex. Remarkably, remember I showed you the A1, the V1, the S1. All of those prominent peaks are into the cortex. And when we look at the sensor level, now we just do EEG. We don't even have to do source localization. It's a very reliable evoked response. And I know, oh, that peak, that's the peak that goes in. So if a peak goes in the other direction, it goes out. We did the same thing with auditory cortex. If we're at sensor level and that peak, I know this peak at 70 milliseconds or 100 in auditory is inward, then any deflection in that direction would be inward versus outward. But that's a really good question and a really important piece of information when you want to connect to circuit level interpretation.
While we're waiting for our next question, I actually have a virtual participant who snuck in through text message because the virtual questions are disabled for this session. But so you were mainly talking about sensory information, and I guess they were wondering, can it be any sort of information that becomes gated or inhibited through these types of beta events that you were talking about?
Sure, it just has to be information that's going to be relayed to that part of the brain. How it gets there, whether it's a thought about moving, right? And so you're sending a premotor signal to the motor cortex, that will come through that same supragranular channel. It doesn't have to be driven by the sensory. in some of these higher order cognitive tasks, there's a lot of attentional mechanisms that are sending communication along different pathways in the brain. You just start with a theory about where is the input coming from and which layer is it going to, and then how can that create a waveform.
Hi, thank you so much. That was a great talk. You made it very clear.
Thank you.
I'm wondering if anybody has looked at the beta event in the reversed way, in that people that are hypersensitive to auditory or tactile, do those people maybe have less beta events?
There are some studies, that autism study that I mentioned looked at beta oscillations. It didn't quantify them as events, because that's a newer line of research in the field, but I'm trying to remember the direction that went in, but likely it is, if you have decreased beta, you would be more sensitive to tactile sensations. But I honestly don't remember the details in that study.
Okay, thank you very much.
Hi, thanks also for a great talk. My question relates to, as it appears to me, you have a model of the cerebral cortex, but there are other sources of electrical activity you can measure on the surface, including from the brainstem and including the cerebellum, which is of interest to me. The cerebellum has a radically different cellular structure, and I would guess that it would require a radically different architecture to model that. I wonder whether you, and for example, one of the things that features of the electrical signal coming from the cerebellum is its predominance of very high frequencies, well above the gamma. And I just wonder whether you had considered applying this to other parts of the brain, including the cerebellum.
Yeah, certainly you can record signals from the cerebellum, brainstem, maybe some deeper structures. HNN is not designed right now to study those signals. And what we're doing is we're relying on our inverse solutions to isolate contributions from the cortex, right? And so we're building, the model is of cortex, and so we can only study cortical signals with our model. And so we're focusing on brain areas that when you apply your inverse solution techniques, we're looking at regions of interest from the cortex. And right now it's not designed for cerebellum or brainstem. As I mentioned, we're trying to make it modular and expandable using best practices in open source software. So if there's somebody out there that's building a cerebellum and wants to integrate it with our framework for studying current dipoles, that would be really exciting. But right now, that would require a different structure in the model.
I had a question too. Can you comment? I'm sure you're aware of Earl Miller's work in the with the relationship between beta and gamma dynamics in working memory. Mm-hmm. Can you comment on how that relates to what you think the general function of beta is?
Yeah, in the memory literature, if I recall correctly, beta rhythms are supposed to be distinguishing the memory and allowing.
I think when you no longer need it, it becomes kind of, that's when you see the beta.
That's when you see the beta.
Right, so I think that's,
yeah.
And there's no, as far as I know, there's no circuit dynamics that are purported to be underlying that, so I wonder if you could speculate.
Well, you could imagine it involves inhibition in a meaningful way, right? If you're no longer wanting to use that information, and so you would have needed that process, but that's just a high-level prediction about what might be happening. And I should say that we're not saying this is the only beta mechanism out there. I know from prior work that there are other ways to get beta oscillations. And so this tool hopefully is designed in a way that you could test other theories and see if they're accounting for frontal cortex beta during memory processing. I think the structure is there. I think the theory can expand, but I'm in no way saying this is the only type of beta there is out there. So that's important. And this transient nature of beta, when you do things like hold something tight, you can see more continuous beta oscillations. And so is that the same mechanism? Well, maybe it's this processing happening in successive time points and creating a more continuous oscillation. Or maybe there's some other phenomena that gets recruited when you're holding something tight that reflected as beta. So, yeah, my intuition is that it probably ports over to what's happening in memory processing, but I haven't studied that.
All right, unless there are any other questions, let's thank our keynote speaker. Thank you, Dr. Jones.
The last thing I want to say is we are eager for the community to use it, and we have a team that's helping support it. So please check it out, and if you have any questions, be in touch with us.